Hi, I'm Steve Palumbi, and this is lecture 10 of the Science of the Extreme Life of the Sea, where we're going to talk about the coldest parts of the ocean, places that are so cold that the organisms that live in them have to have special adaptations to deal with such extreme conditions. So we'll start in the Antarctic with this glorious picture by Jonathan Weller. What you see here is the bottom of the Antarctic Ocean, about 60 feet deep. What looks like clouds on the horizon there are actually the ice up above on the surface. And down below, what look like these white organisms growing up from the bottom are actually ice crystals. And the reason for that is that this water is colder than freezing. Normal water, fresh water will freeze at 0 degrees centigrade. This water is -1 degrees or -2 or 3 or even 4 degrees. And at those cold temperatures, the water isn't freezing solid because it has salt in it, but it is cold enough for ice crystals when they form to slowly grow and begin to take up all this space and look like organisms growing on the bottom. Well, other organisms living there have to deal with the ice crystals that are going to grow within their bodies. These critters, the sea stars here, solve that by having their blood supply, their internal fluid, the same salinity as seawater, so it doesn't freeze at these temperatures. But fish have a problem. Their blood is the same salinity as ours. It's about a third or a quarter the salinity of seawater. And our blood and their blood would freeze at these cold temperatures in the Antarctic Ocean. So, in order for these fish to survive the winters, and they do very well because these kinds of fish make up about 90% of the fish fauna in the Antarctic, they have to have an adaptation to keep ice from forming in their blood. And that adaptation is an antifreeze protein. It's the most abundant protein in their blood, much more abundant than hemoglobin is, which is the most abundant protein in our blood. And the ice antifreeze proteins in their blood, if ice crystals form, the antifreeze proteins stick to those ice crystals. They coat them, shown in this little diagram, and then keep them from forming. Well, that antifreeze protein was strange enough, so people have gone in and looked at the genes and the proteins and tried to understand exactly how it works. It was essentially evolved from an existing protein, a trypsinogen, and it evolved to have a particular set of amino acids that are particularly good at holding on to carbohydrates. Those carbohydrates then are particularly good at attaching to ice. This just shows the backbone of the amino acid chain with some of the sugar molecules that are attached to it in this antifreeze protein. And then this shows how that backbone with the sugar chains here that attach to the lattice of the ice crystal itself. Ice, because it's formed of water, is a very charged kind of structure, has lots of electrical charges on it that the carbohydrates can stick to. So essentially, the antifreeze proteins form a layer of carbohydrates over the ice and then a layer of proteins over that that effectively insulates the ice from the rest of the blood supply. Well, the fish that have this kind of adaptation are called notothenioi fish. They're shown here in their phylogenetic tree with a whole set of different species that inhabit the Antarctic. Their relatives also inhabit other parts of the Southern Ocean. These are the ones that are adapted to incredibly cold temperatures. They also have other adaptations that are that denote their normal habitat of very cold water. Particularly in this case, what's shown here is that this little green dot shows where in this phylogenetic tree this group of fish have lost the ability to respond to heat. There's something called a heat shock response that we have, that most organisms have. If you heat up us or other organisms beyond a certain point, we have a physiological reaction that turns on a whole set of proteins called heat shock proteins that help us deal with the extra heat. This entire set of organisms have not only evolved antifreeze proteins, but they've lost the heat shock response, meaning that they've lost something that they no longer need because they're never really subject to large amounts of heat. Well, as people started looking at the antifreeze proteins in Antarctic fish, they discovered them in other fish and other polar areas, and also they're found in insects. They're found in plants that live in polar areas that are subject to the freezing conditions as well. In particular, some of the Arctic fish that show the same kind of structure. And this is the cutest picture of an ugly fish that I've ever seen. This kind of fish, the eel pouts of the Arctic, also have been studied for their antifreeze proteins. And in particular, the genes from the antifreeze proteins in these species have been isolated and used to create antifreeze proteins in the lab that are then used actually as antifreeze in a number of different lab and commercial uses around the world. It looks like then that both the Arctic and the Antarctic fish have evolved the same proteins, but study of those proteins themselves show that this is a case of parallel evolution. That is, evolution that has produced the same type of product with a different starting material. And we know that by looking at the structure of the genes. This is a structure of Antarctic and Arctic ice or antifreeze genes. Gene structures have introns that are the coding regions that make the protein. Exons make the coding proteins, then introns between them, and the intron-exon structure of a gene would be the same if it had the same evolutionary origin. The intron-exon structure of these genes are quite different from one another, showing that they in fact came from different evolutionary origins. But when you start to look at the sequences of the proteins themselves after evolutionary adaptation, they seem very similar because parallel evolutionary processes have given rise to essentially the same solution from a different starting point. So these fish in cold zones then have antifreeze proteins that coat ice crystals and keep them from growing too much, but they can't keep them from forming perfectly, which means that the blood supply of these fish is essentially floating around with ice with protein coats around it. What happens to that over time? Two things can go on in the bloodstream of these fish over time to sort of rid the fish of these ice contaminants. One is that they're filtered by the spleen, which attaches to the protein and then pulls the ice crystal along with it. But the ice is still there in the spleen and is still there in the blood supply, the part that can't get filtered by the spleen. So it was thought basically that the idea here was that the ice proteins, the antifreeze proteins, would attach to the ice crystals. They would essentially all be stored in the spleen until summer came, and then summer conditions would allow the ice to melt and the system would regenerate. But it turns out that we still don't know the real answer to this because the ice proteins, the antifreeze proteins, actually stabilize the ice as well as coat it and get it out of the blood supply. So if you look at the blood supply of these fish and you look to see essentially where ice melts in normal fish blood, it's at about -1 degree and -1 degrees Celsius. If you then add the antifreeze proteins to it, and those ice crystals are coated, then they're stabilized, and it takes a couple extra degrees, 2 or 3 extra degrees, in order for those ice crystals to melt. That would mean that the summertime temperatures might not get high enough in order to melt the ice crystals that are accumulating in these Antarctic fish. So a set of aquarium studies were done that set the aquarium temperature to be 0 degrees down to -1 to -2. The blood supply of these fish with their ice crystals was tested in these different conditions, and the fraction of the remaining ice crystals was pretty high up until about -1 degrees. It took actually almost to get to -2 or 0 degrees in order for most of the ice in these particular experiments to begin to melt. That means that once formed, the ice is a little bit more resilient than you might think to being melted by high summer water temperatures. Is that a problem for these fish? It turns out that in a number of cases it is. What's shown here are the temperatures in the Antarctic in the year 2000, 2009, and 2012, where the temperature basically got above -1 degrees a bunch of the time. But if it has to get up to 0 degrees, which is up here on this diagram, that it never got there in those 3 years. In what case this means that the adaptation that we know about in these fish to keep them from freezing in the winter essentially is preventing the ice from being scrubbed, from being melted off in the summer by being attached to that same set of proteins. Honestly, we don't have any idea yet how these fish are solving this particular problem. Stabilizing the ice and keeping it growing in the winter is a great adaptation, but if it leads to accumulation of ice over the entire course of the animal's life, we don't know. We don't know yet how they solve that particular problem. If you go back in time and say, well, maybe all they have to do is melt the ice every decade or so because they're long-lived fish. This is a record of the temperatures in the Antarctic over the last 15 years or so, and it seldom, if ever, got close enough to this ice melting temperature for that to be the solution. In part 2 of the lecture on the coldest parts of the ocean, we're going to worry about a different kind of adaptation that animals have. That's not deep sea fish with ice proteins, but this is marine mammals and how they manage to live in cold water, particularly sea otters. When you're a mammal, you have to maintain a warm body core. And if you're a mammal in water, especially cold water, then you need a lot of insulation in order for you to maintain that warm temperature. And blubber is the usual way that marine mammals accomplish that. So in pinnipeds, that is, say, seals and sea lions, there's 7 to 10 centimeters of blubber that insulates them from the surrounding cold water. In whales, the blubber can be 1 or 2 feet thick. That is why whales were so valuable in whale oil hunting, because they basically could, their whole skin and blubber could be peeled off and boiled down to make whale oil. And the more Arctic or the more polar the habitat is for whales, the thicker their blubber. So bowhead whales in the Arctic have about a 50 centimeter or half a meter width of blubber. Smaller cetaceans like porpoises have smaller blubber layers. They're more active, but all of them have a way of insulating themselves from cold water. But otters are different. Otters have only become marine critters in the last couple million years. Their closest relatives, sea otters, closest relatives are river otters. And by contrast, cetaceans have had tens of millions of years, about 50 million years, to adapt to the ocean. Pinnipeds, seals, and sea lions have been fully marine for about 60 million years. Otters are so new at being part of the marine world that they lack a couple of the major types of adaptations that other marine mammals have. Dive responses, that is, when they go down there, they don't reduce their oxygen consumption like most marine mammals do. But particular, they don't have blubber. Yet they live in northern realms in the Pacific Ocean in cold water. So the question is, how do they manage to stay warm even without blubber? So there are 2 ways that sea otters use to basically keep their body temperatures high. One is fur, and the other is a very, very high metabolic rate. This particular diagram shows on the top the fur density. This is the hairs per square millimeter for fur seals and sea lions and walrus. And as the fur density goes down, the blubber goes up. Well, otters don't have blubber at all, and so what kind of fur density do they have? In fact, the fur density of otters is far higher than the fur density of fur seals. They have they have the densest fur of any marine mammal, over 700,000 hairs per square inch. And that has made them actually quite valuable as a target for fur hunting, but it allows them to insulate themselves from the water. So thick is their fur, actually, that the fur itself holds on to air. So when the sea otter is diving, essentially is covered in fur and a coat of air around it, held in place by these thick hairs of the fur, to keep the animal from essentially being too hit directly by the cold water around them. The other aspect of sea otters that makes a big difference is their metabolic rate. This is the metabolic rate of sea otters over time with different kinds of activities, just resting, foraging, grooming, surfing, and swimming. And their metabolic rate is far higher than other animals their size. And that is a way for them to generate metabolic heat to keep themselves warm in cold water. In fact, the basic maintenance cost, the basic metabolism of a sea otter keeping itself warm is the biggest metabolic cost it has. This is the metabolic cost in sea otters that are eating different types of prey, crabs, mussels, or mixed prey. And the black part of these bars is the amount of energy they spend just maintaining their normal body temperature, and it's 2/3 or so of their entire metabolic rate, compared to the cost of locomotion or even foraging, much lower than the costs of just staying warm. Well, as a consequence of that, even though they have thick fur, otters basically maintain their body heat by eating, and they eat a lot. They eat a lot of different kinds of prey. They're constantly foraging. In fact, an otter will generally eat about a quarter of its body weight a day. So a normal-sized otter might be 60 pounds or so. That means it's eating 15 pounds of seafood a day just to maintain its body temperature out there in the cold water. And as a consequence of that high amount of food consumption, otters play a very, very, very strong role in the ecology of the areas that they are, particularly in kelp forests. This is a classic study done in the early 1970s of the relationship between otters and sea urchins, one of their favorite types of prey, and kelp forests that they live in. And what's shown here is the test diameter, that is the shell diameter of sea urchins in areas that have otters and in areas that do not have sea otters. These are in Alaska. Where otters are common, urchin populations are quite a bit lower, and the individual sizes are very much lower. Where there are no otters, then sea urchins tend to bloom in abundance, take over, and become quite numerous and quite big. Without otters, then you get many, many, many sea urchins. The sea urchins consume the kelp, and the kelp forest is much thinner, actually disappears in a lot of places. With otters, then the sea urchins are consumed. The sea urchins don't consume the kelp, and you get a large stash of kelp plants growing. So this is called a trophic cascade, where a predator eats its prey, and the prey itself then controls the abundance of, in this case, the plants that grow up and form the basic ecosystem. And this trophic cascade means that sea otters are a keystone species in these habitats because their presence or absence totally changes the habitat around them. In the late 1990s, Jim Estes and his colleagues also added another element to this trophic cascade, which is orcas or killer whales. In Alaska, the presence or absence of killer whales or their propensity to eat or not eat otters then adds another layer where orcas consume otters, then sea urchins become much more abundant and the kelp density drops. Where the orcas don't consume otters, then the sea urchins are eaten by the otters. The sea urchins don't eat the kelp, and the kelp forest blooms. So there's a trophic cascade that relates killer whales to marine algae in these habitats, and changes the nature of the habitats depending upon what the orcas are actually feeding on. Humans also play this kind of role. When otters were first started to be hunted in the late 1700s in the North Pacific by Western traders, those otters were then sent into fur markets mostly in China and the rest of the world. Then a large number of otters were hunted in Alaska, down through the coast of North America into California, and even in Baja, Mexico. We know just in Central California that records of ships leaving with furs for the Chinese trade had almost 40,000 otters pelts across the years from about 1789 to the early 1800s. It's estimated that about 100,000 otters were killed and then placed in the fur market in that period of time. And what that led to was a dramatic decline in otters all along the West Coast. In fact, they were considered to be almost extinct along the west coast of the United States. And that led to a bloom in sea urchins, a bloom in abalone, which is another thing that otters eat a lot of, and then a huge decline in the kelp forests along the west coast of the United States. So just otters staying warm means that they're eating a lot, which means they play a really strong role in the ecology, which means that things that control otter abundance have an outsized role in the ecosystem of the kelp forests of the west coast of North America. So what other kinds of cold-related problems do predators have, and what kinds of other ways of adapting do they have? There are other predators in the ocean environment that are also affected by the cold that's there. And in particular, it turns out that other predators like polar bears have strong diet needs as well, and have to eat a lot in order to survive the cold. This particular study shows this by essentially following a set of 9 different polar bears during a short period of time as they're foraging and hunting and also growing. And so for the first time, we could see what their activity patterns are, how that leads to their ability to add layers of fat before hibernation, and exactly how much prey that takes in order for them to succeed. This particular diagram shows the movement of individual polar bears across the landscape. They were captured, fitted with satellite transmitters that allowed them to be tracked. And then at the same time, the investigators could also look at their mass change by weighing them before and after, and also what they managed to feed on. Well, part of the reason for concern is that polar bears have a pretty high metabolic rate for their size. This is a diagram of body mass versus the resting metabolic rate. Different kinds of bears, including some pandas and a sloth, and polar bears are high up on the metabolic rate here. They also are required to build up body mass at the end of the summer in order to hibernate for the winter. And in this particular set of studies, it turns out that among the 9 polar bears that were followed, 4 of them actually gained weight, but 4 of them lost weight. One of them just basically stayed about the same. So these animals, these ones that have gained weight, are probably in pretty good shape, but losing weight before hibernation is a bad sign for polar bears. How much prey does it take to actually add weight if you're these animals? In this period of time, some took no prey at all. Some took one, two, or up to three different captures across this particular study. In particular, the animals that gained the most mass had the most prey that they captured. Telling us that, in fact, the prey capture rate is a really important part of the ability of these animals to have a high metabolic rate, add mass, and then be able to hibernate in the winter. Part of the problem is that sea ice has been declining in the in the Arctic over the last decade or so, meaning that the foraging area that these polar bears have is getting smaller and smaller and smaller. Their access to prey is getting lower and lower and lower. And so you take a high metabolic rate, a high need for a large number of prey taken in order to gain mass to survive the winter hibernation, and then take all those needs and begin reducing the foraging capacity that these animals have because sea ice is breaking up. And you set these animals up for an ecological crisis where they simply are, it's too cold, there's not enough prey, and there's not enough foraging in order for them to be able to survive their winter hibernation.